Chapter 25 discusses or respectively just introduces population genetics. It is a branch of genetics that studies the genetic makeup of group of individuals, genetic makeup of populations. So now look at these two pictures. You have population of humans and population of ladybugs. What you can see immediately is that both populations have extensive variability. There are no two people which are identical. So this variability is based of the evolution, and what determines this variability are the genes which makes these populations. So this variability and changes over the time. And what population genetics study is not just genetic makeup of groups of individuals, but also how the genetic composition changes with the time. Studying that, population genetics usually focuses on so-called Mendelian population, which is defined as a group of interbreeding, sexually reproducing individuals that have a common set of genes. This common set of genes forms so-called the gene pool. And population usually evolves through the changes in its gene pool. So therefore, population genetics actually is base for studying evolution. So let's now talk about what defines the structure of the genetic populations. And basically it is the frequencies of genotypes and alleles, so genotypic and allelic frequencies. So before we go to Hardy-Weinberg model and how do we use it to define the population and frequencies of particular genotype or phenotype, we will actually focus on how to calculate genotypic and allelic frequencies. First, calculation of genotypic frequencies is relatively simple. You divide number of individual with particular genotype by total number of the individual in the population. For example, if you calculate frequencies of homozygous dominant individual or genotypes, you divide the number of homozygous dominant by total number of individuals. If the same apply if you calculate frequencies of heterozygous, where you plug in number of heterozygous and divide it by total number, and equally for homozygous recessive. You take number of homozygous recessive in the population and divide it by total number. Now, allelic frequencies can be calculated by two ways. They can be calculated from numbers or of the genotypes or the frequencies of the genotype. If you calculate it, firstly we will calculate it from the number of genotypes. So when you calculate allelic frequencies from the number of genotypes, be aware that each genotype, each gene has two alleles. So if you, for example, calculate the frequency of dominant allele from a number of individuals with dominant phenotype, which are homozygous dominant, you have to multiply it by 2. So let's say that we define that P will be our dominant allele and Q will be our recessive allele. So if you want to calculate frequency of the dominant allele, which equals frequency of the A, you take 2 times number of homozygous dominant plus number of heterozygous and divided by 2 times total number of genotypes. Remember, each genotype has 2 alleles. The same apply when you try to calculate the frequency of the recessive allele from the number of genotypes. You multiply number of homozygous recessive by 2 because there are two alleles, plus you add number of heterozygous which have only one recessive allele and divide by 2n. Now, if you want to calculate, if you allelic frequency from genotypic frequencies, respectively, if you know genotypic frequencies, it's very easy to calculate allelic frequencies. So if you want to calculate frequency of dominant allele and you know genotypic frequency, so frequency of dominant allele would be frequency of dominant genotype plus 1/2 of frequency of heterozygous. The same for frequency of recessive allele, where it will be frequency of the recessive genotypes plus 1/2 of the heterozygous genotypes. Later on, we will also discuss how to calculate frequency of allele with multiple allele in one locus, and also for XY chromosomes for X-linked genes. One of the most important principles of population genetics is Hardy-Weinberg law, which was formulated at the beginning of 20th century. Hardy-Weinberg law is actually a mathematical model which evaluates the effect of reproduction on the genotypic and allelic frequencies of the populations. We know that primary goal of population genetics is to understand the process that shapes a gene pool of the population. And the question which population and genetic ask is how segregation of alleles during gamete formation and the random combination during fertilization influences the gene pool. And Hardy-Weinberg law can give the answer to these questions. And since it is a mathematical model, it has to actually be simplified, and it has to be based on simplified assumptions. So Hardy-Weinberg law only will apply if following assumptions are met. Population is large, randomly mating, and is not affected by migrations, mutation, or natural selection. If all these assumptions are met, and we can say that the population is in Hardy-Weinberg equilibrium, and we can predict. We can make prediction that the allelic frequencies of the population will not change, and the genotypic frequencies will stabilize. So what does this law indicate? It does indicate that when all the assumptions are met, reproduction alone cannot really alter allelic or genotypic frequencies. So reproduction alone cannot introduce variability and push the evolution. So genotypic frequencies usually stabilize after one generations. So during that first generations they can change, but afterwards the genotypic frequencies, similarly to allelic frequencies, will not change, and populations will actually settle with a genotype as expected, as we will go over, p square, 2pq and q square. And if this happened, we said that Hardy-Weinberg equilibrium was achieved. So how do conditions of the Hardy-Weinberg law led to the genotypic proportion p square to pq square, and what do they mean? So let's first talk about what P and Q and this P square, Q square, and 2PQ means. So you remember in previous slide we said that we will assign P corresponding to the dominant allele and Q to recessive allele. So if you have a genotype, this genotype, even so there might be multiple alleles, but in one locus, in one gene, you can only have two alleles. So if you have two alleles, so sum of this allele is 100% or 1. So P plus Q will always equal to 1. P square plus 2PQ and Q square therefore equals 1. So what is P square? P square are homozygous dominant allele pair. It's a frequency of homozygous dominants. Q square is frequency of homozygous recessive allelic pair or genotypes, and 2PQ is frequency of heterozygous genotype or allelic frequencies. So how did Hardy-Weinberg law led to this genotypic proportion or frequency of this genotypic proportion? So if you consider Mendelian population with random mating, so you have allele which are dominant and allele which are recessive. You remember we assign P to dominant allele and Q to recessive allele. If the mating is random, so basically gametes will come together in random combination, which can be represented by Punnett square right here. And basically when there is random mating, random fertilization follows. So basically the gamete with dominant allele can be fertilized by gamete with dominant allele, so you get P by Q, which is P square, which represents homozygous dominant. However, the same gamete which contain dominant allele can be fertilized by gamete containing recessive A, and you have P by Q, which is heterozygote PQ. The same here, recessive allele can actually be fertilized by gamete with dominant, and you have PQ. So if you put them together, you have 2PQ. And if two gamete containing recessive allele fertilize, you will get homozygous recessive or Q square. And basically, by applying these multiplication rules, you find the probabilities that these gametes will combine and produce these genotypes. So, if we summarize this, whenever there are frequencies of allele in a random mating population are P and Q, the frequencies of genotype in the next generation will be P square for homozygous dominant, 2PQ for heterozygous, and Q square for homozygous recessive. You remember the genotypic frequency stabilized after one generation of random mating. So what are the implications of the Hardy-Weinberg law? Firstly, we know that if the population is in Hardy-Weinberg equilibrium, allelic frequencies do not change and genotypic frequencies stabilize after one generation. So the implication of this is that population cannot evolve. It cannot evolve if it meets the Hardy-Weinberg assumption. Why? Because evolution needs change, needs variability. It needs change in the allelic frequencies in population that there is a base for this selection. So Hardy-Weinberg equilibrium means that it cannot evolve. Respectively, population which is in Hardy-Weinberg equilibrium cannot evolve. Now, when population is in Hardy-Weinberg equilibrium, the genotypic frequencies are determined by allelic frequencies, and single generation of random mating produces equilibrium frequencies of P square, 2PQ, and Q square. As mentioned previously, if a population is in Hardy-Weinberg equilibrium, the genotypic frequencies are determined by the allelic frequencies. And the implication of this for heterozygous is that if you look at a locus with two alleles, the frequency of heterozygous is greatest when allelic frequencies ranged between 0.33 and 0.66, and the maximum of heterozygote in population are achieved when allelic frequencies are equal, when each one is 0.5 right here. And it makes sense. Think about that. If you have equal number of gametes carrying dominant allele and equal with gametes carrying recessive allele, it's higher chance that a gamete with dominant allele will meet gamete with recessive allele. Opposite to it, when the frequency of one allele is low, of course, homozygous for that allele will be rare, because if you have very few gametes with that allele, there is very low chance that these two gametes carrying this rare allele will meet. So the Hardy-Weinberg expected proportion of the genotypic frequency can be also calculated if there are multiple alleles in the locus, and also when it comes to X-linked alleles. So let's consider that you have locus with three different alleles, so two alleles can be selected out of the three. A1, A2, and A3. If you know frequencies, let's label frequency as A1P, A2Q, and A3R. So we can also calculate genotypic frequencies for these three alleles by formula square of P plus Q plus R. And when you calculate this, it is outlined right here. So what would be the result? It would be P square plus 2PQ plus Q square plus 2PR plus 2QR and plus R square. So you can extend the same way equilibrium expected proportion of the genotypes if you have 3 or 4 alleles. Now, when it comes to X-linked alleles, of course you know that if allele is X-linked, female are expected to have two alleles, either 2X1, which are P square, either 1X1 and 1X2 2PQ, or X2 2X2, which is Q square. Let's say that the X2 is recessive, so homozygous recessive in this case are Q square. Now we know that male carry only one X-linked allele, so the X2 will be only Q under this situation. So what is the implication of this? Let's consider certain disorder which is X-linked, such as hemophilia, which is disorder in the clotting. Let's say if 1 in 10,000 people carry the allele, so frequency of Q is 1 out of 10,000. So basically a Q frequency is 0.0001. So this would be frequency of the disease amongst the male. But how does it apply in female? So in female it would be 0.0001 square, which would come up to 0.000, oh my gosh, 0.0001. So you see automatically there is much lower frequency of possibility of the disorder appearance of the disorder among female. We know that female can be carrier, but frequency of disease such as hemophilia is higher in the male as in female. So, what is the practical use of Hardy-Weinberg law? Well, as you could see with hemophilia, it allows to calculate allelic frequencies if there is dominance present in that trait. So what implication it has? We can actually calculate frequency of carriers, which allow us to predict number of potential people which would suffer of the disorder. One of the assumptions of the Hardy-Weinberg equilibrium is random mating. However, many populations do not mate randomly, and non-random mating will affect the way in which alleles combine to form genotypes, and non-random mating will alter the genotypic frequencies of populations. So what are the forms of non-random mating? It can be positive assortative mating, which is a tendency of like individuals to mate, such as, let's say, tall person likes to mate with tall person while short person likes to mate with short person. An opposite to it is negative associative mating, which is a tendency of unlike individuals to mate. Let's say tall person prefer to mate with short person. One form of positive associative mating is inbreeding. Inbreeding is mating between related individuals. This leads to an increase in the proportion of homozygous in a population, and decrease in heterozygous and decrease in variability. Now, inbreeding has very strong impact on the population. Usually when you have small isolated population which inbreed, you can see much higher frequency of the disorders which are encoded by recessive alleles. And it is because the number of homozygous are increasing and number of heterozygous are decreasing. Now, the avoidance of mating between related individuals is outcrossing. We talked about inbreeding, that inbreeding increases number of homozygous and decreases number of heterozygous. So if there are homozygous in the population, does it mean that these homozygous are a result of inbreeding? Not necessarily, because these alleles, which are, let's say, allele A1, which can be some allele which is recessive and carries certain disorder, can actually form homozygous. These alleles, two alleles which you can find here, can actually be identical by descent, where it indicates inbreeding, but they can also be identical by state, where there is no inbreeding. So what does it mean identical by descent? Let's say several populations or several generations before, some person actually, in some persons, this allele underwent mutation from A2 or A3 to A1. Now, this person, of course, transfers this allele to offspring, they transfer it to offspring, and so on, and these two offsprings actually made, there was inbreeding and brought these two allele A1 together. So these two copies are descended from the same ancestor. So the same ancestor give rise to these two copies, so that these two alleles are identical by descent. However, in this case, this individual, which also carry two A1 alleles, in this case, these two alleles are identical by structure and function, but are descended from two different copies in ancestor, so they are not identical by descent but by state. Why does it matter? Because we can actually measure inbreeding, and inbreeding is measured of as the probability that two alleles are identical by descent, right here. Now, inbreeding is measured by inbreeding coefficient, which is F, and it can vary from 0 to 1. If inbreeding coefficient is 0, it means that there is no inbreeding in the populations. However, if the inbreeding coefficient is 1, what does it mean? It means that alleles are identical by descent. And you can calculate inbreeding coefficient from the pedigrees. And it's very important because a level of inbreeding actually can tell you probability of increased frequency of homozygous in the population. One of the way of inbreeding is so-called selfing. It is self-fertilization which certain plants undergoes. Selfing will reduce the proportion of heterozygotes in the population by half with each generation. You have it outlined right here. You have number of heterozygotes in first population 1/2, afterwards next one 1/4, 1/8, and so on. So for most species, this close inbreeding on selfing is harmful because it increases the proportion of homozygotes, and thereby it increases the probability that lethal or deteriorous allele will increase. This increased appearance of lethal allele or lethal traits is termed inbreeding depression. And the more intense the inbreeding, the more severe the inbreeding depression will be. There are certain exemptions in some crop, in some plants, where actually inbreeding is not deteriorous, and it is because those with harmful allele were weeded out by natural selection, and basically only beneficial allele states. So homozygous is beneficial traits. However, it is beneficial until this population is not exposed to certain environmental stress, let's say some new pathogen and so on. Because it is homozygous, it has less variability to survive stress selection. So this is just summary of what we were already discussing, that inbreeding increases the percentage of homozygous in population. Selfing reduces the proportion of heterozygous by half each generation. You recall we had a table which outlined it in previous slide, and this leading to a completely homozygous population. After so many generations, heterozygous would disappear and population would be homozygous. So mating between siblings increases the percentage of homozygous relative to random mating, and mating between cousins also increases the percentage of homozygous, but in slower rate. It's like here. Basically, you see siblings share much more similar genes, so they have approximately 50% similarities. So mating between them increases the percentage of homozygous right here, and in first cousin right here it would also increase number of homozygous, but in slower rates. And it's why this was actually incorporated in many laws in the population and societies without really knowing genetic base for it. But you know that inbreeding, mating between or marrying between two siblings was forbidden, because this was only based on observation, but it was because that observation showed that it is harmful for the offspring and for the population. And similarly, inbreeding has very often deleterious effect on crops. This is in corn where inbreeding coefficient, with increased inbreeding coefficient, you see that average yields goes down. And if you come to inbreeding, the yield is very, very low. We already discussed the exemption when basically homozygous are beneficial. There are some exemption as that, but in most cases, inbreeding is harmful also for crop.